Okay, so let's get going, everybody. So it's my pleasure to chair this session on new dimensions to enable new questions, which is on reflection, neither a grammatically terribly coherent nor scientifically terribly coherent title for a session, so I didn't spend an enormous amount of time thinking about that, clearly, on reflection. Nevertheless, I do hope that the next hour and a quarter or so are going to be fun. We have 3 of us doing this, okay? So, okay, so we have Aviv, whom some of you know, and Dana online again, who unfortunately, despite 3 attempts, has been foiled by the US airlines and 5 attempts, oh my gosh, 5 attempts to make it from New York to Toronto, and the failures of US airlines. So I was ready to make various comments about you weren't hallucinating, but there would be actually a Dana in person, but no, there is still only a virtual Dana on the screen, I'm afraid, no in-person Dana, but nevertheless, really looking forward to this. So I hope that this is going to be a really interactive discussion today, and that it's going to touch on topics that are really of interest to this entire community, because inevitably a lot of discussion at HCA meetings over the last, 6 years since the first one, 7 years, 2016 in London 2016. Was the first meeting in the Welcome. Much of that conversation has been about how to build an atlas. But we're now at the point where really we should begin to think about how do we interrogate it, both with and without independent data sets that are not part of the atlas. And I think that will help us think about the sorts of tools that we need to develop to interrogate the atlas and really how we're going to use it. So the focus of the conversation today is really on that. So to start, we'll start with something very simple. Let's think about the types of questions that you might want to ask of an atlas in the context of a particular biological question, but without a separate data set. And I would just give a couple of very simple examples. In which cells is my gene of interest expressed? In which cells is my gene module active? These are kind of obvious questions. Actually, it's not entirely trivial at the present to answer that question with the way that the data are presented and available. You might be able to do it for sub-data sets, but doing this at a bigger level, harder. But nevertheless, I would say those are some of the more trivial questions. But now let's go to more complex questions. Where is a specific molecular gradient present? So see, I've got a whole human cell atlas across many tissues, and I want to know where there's a gradient of expression that's driven by Wnt signaling. Would I be able to interrogate the atlases as currently constituted to find that? Again, that would really be very non-trivial, I think. But these are the types of biological questions that one might well want to ask of an atlas. And so I think that it really is incumbent on us as a community to start thinking about this, because it's going to help us structure the resources that are going to be available and how we can develop the statistical tools to answer some of these questions. So to begin this discussion, and then we'll turn it over to the floor, I'd like to ask Aviv and Dana to propose some of the questions that they would want to ask of an atlas, not with an external data set, but just the type of biological questions that they would like to ask of the human cell atlas in its entirety. And we'll start with Aviv down at the front here.
First, I just want to, I'm supposed to use this. Sorry. I just wanted to say it's important to understand why the questions that you said were trivial were actually not trivial at all. So for example, when you say, in which cells is my gene expressed, you make a lot of assumptions. For example, you make an assumption that there is a category of cells that you want to go after. But maybe the person asking the question wants individual cells. Maybe they want a group with a label. So actually it's embedded in the question, but unless you define it, it can be a hard or an easy thing to do. And unless your cells are organized in a certain way, the answer is not well defined. And the same is true for the word expressed. Most people, when they use that word, they mean relative to something else, like a lot more than somewhere else, or at a higher level, or more specifically, or not. It's just that the queries, these questions are really easy to say in English, but then when you try to make them into a something that's executable, they become, you realize that they were ill-defined. Now, more to your original question, I'll suggest a couple. One is, what are the dependencies between my cells? So when when these cells look like this, what is happening to those cells at the same time? I think that kind of pattern is interesting. We don't ask about very frequently. And the second one is, how do the relations I see between the cells depend on the genes that I'm using in order to look at them, and vice versa? How do the relationships between the genes, since you were referring to gene programs or modules, how do the relationships between the genes depend on the cells that I'm considering? And I'll turn to Dana.
Yeah, I mean, again, I really want to stress that even, your simple questions are absolutely not simple. There's huge technical differences between data sets and even between cell types. And, if you just look globally, how much is my gene expressed? There's differences in just capture, like, macrophages are huge, T cells are tiny. And if you look at something like different inflammatory genes, there's different cell types that have a different baseline. So the question is, like, is this gene expression interesting? Like, if you look at, like, interferon, you'd probably get all macrophages and ignore the whole rest of the immune system. I really think that we've been really focused on too much of a cell-focused view, on labeling cells, on giving cells names, on showing the cells in a latent, which, as Aviv said, is entirely dependent on the features we choose. Even the simple UMAP, it's all dependent on how we choose our highly variable genes or whatnot. And so we really need to start thinking more about the genes and the gene programs, and even how to define meaningful gene programs. I actually think of gene programs as these little Lego blocks that compose cells, and we should begin defining cells as the gene programs and their activity that are active in the cell, rather than, one label per cell type. Now, I like understanding how things form, and, Aviv gave one example of how different cell populations might influence one another, but, we've sort of realized that the cell types have a manifold. Part of this, what we see, is how we chose to represent it. But I really want to begin to understand what are the factors that are giving this structure in the data, what type of structure is driven by spatial gradients, what type of structure is driven by developmental gradients, and what type of structure is driven by all sorts of regulatory forces that are actually giving us this atlas. And once we can begin to understand this, we can understand what goes wrong in disease. And many times, like particularly in a disease like AML, which is a derailment of regulation, the AML populations can look very, very, very different. But if you look at the derailment, sometimes it's very few mechanisms that just send the cells off in a completely different way. So I think we need to begin being able to ask, what are the forces that carve our observed atlas? And how are these forces altered and changed and disrupted in disease? And we need to be able to look at it at the gene level, because genes is what you can target it at the end with drug, rather than these uninterpretable latents where you lose the genes.
Okay. So I think that in those answers or the questions that we've posed, there's at least a couple of common themes. One of them is the ability to answer questions at a gene or at a module level, to understand where is a gene expressed, what factors, what modules are active that are explaining the structure of a particular manifold. And then there's a question about, a second type of question, about dependencies between cells, between how those dependencies change depending upon whether I'm looking at signal ingredients, whether I'm looking at sets of transcription factors, how those interactions change. So those are two classes of questions that Dana and Aviv have come up with on the gene and the dependency level. Now to people here, what other types of questions, when you think about how to interrogate an atlas, are of interest? And we'll start with our host, Gary, down at the front.
Yeah, so I like the idea of modules, but I wonder if we could extend it to more multi-scale thinking. and also think about function. So we've spent a lot of time identifying gene function, have different aspects of gene function, and we've identified, and some of those relate to the modules that the gene functions are in, some of them relate to the cells that the genes are expressed in or the tissues. But what are the other levels of structure that we can look at, and what are their functions? So are there a set of cells that work together to handle wound healing or some other function? And can we identify all of those different functions and their different aspects at multiple scales to and understand how they're related to each other? I mean, I wonder if that's a helpful way of thinking about things.
To me, those are these dependencies, right? So that it is not just each entity to its own, and it can be cells in cellular data, it can be in spatial data, it can be at this scale, it can be at that scale. But these are dependencies by now between those variables. So it can be when this is positioned here, what is happening to what is positioned there? Are these things, dependently positioned together? If I see one of these, I'm going to see one of those. Many, many ways in which you can think about that. That's just if you look at one dimension. Or when I see things at this level of organization, it's dependent on what's happening at another level of organization. That's why I think dependencies are really interesting. In addition to, not instead of the more standard queries of, where is, this is expressed in this entity, this is related to that entity, which is maybe a less exciting dependency.
So we'll come to it in a second, but just, Gary, I'm curious, do you think of this as a hierarchy of modules and cells? Do you need to think of a hierarchy of modules that are present in different sets of cells that are discriminating? Is that how you would think of when you say multi-scale? Is that a hierarchy implied here, or is it just, we took the microphone away from you. That's fine, it's returning. And then we'll go to Dana.
I meant sort of thinking about functions at different scales, like, I need to move my arm, that's a pretty big scale. There's a bunch of cells involved in that, right? Wound healing, that's a smaller scale. There's a bunch of cells involved in that. And then there's, all sorts of different levels all the way down, but it doesn't have to just be cells. There's modules inside the cells as well. So, and we've ascribed function a lot to intracellular systems, but we haven't done as much thinking about all those other systems at all those other scales. I mean, we do know what the function of the arm is, but those are all linked. They're all systems of cells.
So then Dana and then Bruce. Dana first.
So what I tried to say, the gene programs, if they're good, are not just random factors in NMF, but actually a set of genes that are supposed to actually execute a program. And this is, what I sort of began to allude to when I said forces. I think, as we're having other modalities with the attack-seek, for example, we could begin to ask what are the cell intrinsic forces that actually execute a function, a particularly inflammatory module gene program being inactive into a cell. But that's also in the tissue context. So there's intrinsic factors. And I think we're collecting spatial data. We're defining neighborhoods. We're cataloging. We're saying, okay, here's a neighborhood that's defined. We saw an example of this today that's defined by this sort of proportion of different cell types, but we haven't really grappled with, defining neighborhoods as a bunch of cells coming together in a tissue for function. And spatial certainly gives us multi-scale where we have very local neighbors and as that expands and as the tissue is being influenced. And I think we're doing a pretty bad job right now, and something that at least I spend my days thinking about is how do we begin understanding these cellular groups that we are beginning to see together in the tissue in a functional way as a number of cells that are interacting with each other with the multicellular role of the function. So I think this is very central, and it's sort of part of what I said of what are the forces that are shaping this atlas that we're seeing.
Bruce?
I just haven't thought about Dana as an anarchist before, but now it's really clear that she wants to break down everything and then rebuild it. But I think that Gary's answer to your question, John, was yes, hierarchies. But what hierarchies and how, and how to assemble them? I mean, these are like the metadata levels around genes, around cells, around tissue domains, with, around, interacting organs that are going to do something like move Gary's hand. And, I think that we need systems modularization and hierarchies they're practical and, and leverageable at, to do things like have a context-specific query about a gene and about the cell populations in a context, and then, differential context comparisons of population structures or sub-differentiated states or new members of an ensemble. All that stuff should be doable, and I think it's only doable in a sane way by maintaining hierarchies and figuring out what are those hierarchies that we really are. Maybe they're not, they're not real. They're just, constructs that we come up with. Some of it's data driven. Some of it's our own mental machinations that just help us think about it better. But that, the idea of here's a stromal cell that's around a blood vessel element. Here's like a big tube. Here's a very tiny tube. Now these are like just the concept of tube control, and then disruptions of tube structure. Those are concepts that can link all kind of branching or organogenesis type or organs across, so we can get data from the breast to talk to the kidney, to talk to the lung, to talk to other structures, and we get information borrowing across there that really it helps us understand gene networks of a specialized element in that ensemble that's functional. So I think we need to think about what are our hierarchies that are just useful attributes that people can put on about their cell, about their gene, about this this context that they're seeing just function, dysfunction.
So I'm curious about this sort of the hierarchy concept, or the cross-tissue that you say, because I know that, for example, Emma has worked on sort of the cross-tissue early developmental work, right? And the sort of the crosstalk in the immune sense across tissues. But do we think about working across tissues in a whole body context in the sort of the mature cell atlas? Or how do we think about that? Is that kind of fitting a massive model across everything? Is it fitting multiple models per organ and then trying to combine them in some way? How do we more concretely think about this? So we'll start with Aviv and then Serena.
I'll say two things. I think practically you kind of try and do both, but the underlying reason for that is something that we talked about also yesterday, which is that we know already. It's not a new scientific insight. It's just one that we can make we can instantiate really well, that cells are influenced in their state by multiple factors. And two of the dominant factors that we see in multicellular organisms like humans are actually their cell identity, which is defined by their developmental history. And they are a T cell, they are a B cell, they are an epithelial cells. That's what I mean by that. And the tissue in which they reside, which for a parenchymal cell is almost one-to-one, like their development and their tissue residency is kind of the same thing. Neurons live in the brain type of idea. But for all other cells, it's actually not. So, your T cells, your resident Tregs in the gut are not the same as your resident T cells, Tregs in the lung, but they're both Tregs. And that duality is one of the interesting things to parse out. And you don't necessarily parse it out very well if you treat each of them kind of in isolation from each other and then you compare them. But also, if you constantly want to hug like everything, then sometimes it's an overkill compared to the problem that you want to ask. Which is why you need room for both approaches rather than having to choose only doing it one way or only doing it the other way. But we know the signal is there. I mean, there's absolutely no doubt about it. It's a well-established biological fact. It's more about getting the details that we actually need in order to understand it. Now, you can generalize from this point and say there's other factors like that impact the overall status of a cell. There's where it is in the cell cycle and where it is located very specifically relative to other cells and the forces that act on it, the physical forces and the molecular information that it's getting. There's many factors like this, and the combination of them is not linear. And parsing that out, that's a good problem to set ourselves, and that's maybe how you think about the full integrated entity. beyond searching for the genes in the cells.
So Zarina, then Dana.
I wanted to follow up on things that you all said. Dana talking about factors that influence groupings of cells, dependencies, hierarchies, and so forth. And again, ask us all to think about the basic function of every organ is expressed by known types of cell. There's a limited number, 3, 4, 5, depending on which organ you are in. We are talking about things that are stationary. Immune cells can sometimes work solo, but the kidney, the lung, the heart, there is always a grouping of cells that need to work together to perform the function. In order to have enough output, you have hundreds, millions, hundreds of thousands or millions of these units working together. So that's in a way I would think, and I think HubMap is trying to work on that, but I think we all need to think about how to do this computationally and otherwise. How do you represent the basic function of each of these organs by looking at the sort of most basic unit that's needed to do, say, filtration in the kidney, right? So it's a known entity. We know, or at least we think we know what type of cells we have, and you don't need too many of them. So I think that experimentally, computational, you could do that. You could also see changes in function if there is any problem with the organ. That's the place where you would see first. The rest of the structure of the tissue is supporting stroma, bringing oxygen, doing other things. But that unit, yes.
So Dana first, and then Aviv. Sorry, Dana.
So I guess, I'll relate to two of the comments. So, I can't agree more with Aviv in terms of, we really have to sort of break down what's happening into the sort of factors or the forces that are creating this signal, almost, a mechanistic generative model, if we're going to talk about sort of these foundation models. And, in terms of, context is important. Aviv described context, the context of the developmental history, the context of the tissue, and things change under context. But one really, really powerful force, and, I'm actually going to quote Aviv in her first, even in her, fellowship, that evolution is the strongest force, and the cell have reinvented things over and over again. It's one genome, evolution, and reuse of these programs and genes that adapt to different contexts, to different cell types, to different tissues, and I think that because it's one genome and some massive evolutionary force that generated this to change under these different contexts, that I think we have a chance to begin to understand the rules. Now, in terms of spatial role or these functional units in tissues, that's what I said in my previous comment. I think that's the biggest challenge that we have for the spatial field. What are these group of cells doing together? Why do we see this spatial pattern? How are these cells interacting to create a function? And I think here we're actually, one of the reasons we're stuck is we're missing a little bit of data. We have observations of the cells, of their genes, of their spatial location, but we don't have any very measurements of function, of the oxygen, of what this unit is doing. So I think that if we want to get there, we need to begin getting some type of way to assign functions and incorporate that into our learning, because otherwise you see a bunch of cells, you see their pattern, but you don't have much about the actual function at the organism, at the level of an organism and organ and organism.
Okay, so Aviv, and then I think it's actually Partha just behind you, and then Piero, and then in that corner. But Aviv first. So
to go back to Zarina's question, this is what I think of as histological modules. When you have a certain structure and function in biology tend to go well together too, just like evolution is an ultimate signal. But But structure and function tend to go very well together, just like they go, say, structure-function relations in proteins, the same is true in the tissue. So you have a histological structure that tends to repeat itself, which is how you know it tends to repeat across individuals within the tissue. And the molecular and cellular entities that are there, that's a histological module. And now you can define them in one of two ways. You can go the supervised route, because honestly, just like what I said yesterday about CD8 T cells, we say CD8 T cells and say, oh, it gives us functional data, the single-cell RNA-seq. It doesn't give us any functional data. We actually knew the function ahead of time. And we found the molecular correlates, and now we can ascribe the function to that because we actually had it before. So that doesn't give us function directly, but it's a great functional corollary. Here, the same idea applies. There is very rich data from histopathology collected over 150 years or so, and that's the supervised approach to it, to say, these are entities, we know they exist, now we relate them also to all these molecular features, the cells that are there, and that gives us a richer description of that unit. That's approach number one. Approach number two, not mutually exclusive, is the unsupervised approach. It's to say there should be structural entities like that. They would have a repeatable visible representation to the multi-scale world, and they would have in them cells and molecules and so on. And that whole thing should exist there. I don't know what it looks like, but it should exist there. Let me go with an unsupervised approach and pick these things out. The corollary to that would be you can take every cell and score genes you already know expressed in CD8 T cells, and you will pick out the CD8 T cells, or you can cluster and you find a cluster, and then you realize and you label it post hoc. Both are legitimate approaches. You kind of want to use both in this case. And I think if you use an unsupervised approach, recover back things you already know are there, then you start believing the things you recovered there that you don't know yet what they do. And then to Dana's point, at that point, you actually need to have an approach that would allow you to start deciphering what these new things are doing. In animal models, it's a lot easier. You can come and you can start poking and removing things and adding things. In humans, it's always harder. You will be relying on associations, interventions like drugs and treatments and so on, and some human genetics. It always repeats itself in this way. It's not unique to this problem. You have to take the playbook and kind of just twist it for this problem at hand.
So in the middle first.
Hi, I'm Ben. I'm with the IGVF, not HCA. Thanks for. Oh, the other person first?
No, you first.
Okay. So I was just thinking about what Gary and Dana said. That there sort of is a skeletal framework that you could begin to start mapping functions to cells, which is actually the set of Go annotations, right? So we have a theoretically a mapping of genes to cells, and we have in Go theoretically a mapping of genes to functions, processes, etc. Like you could apply those, smash them together in context. You could begin to sort of build this, what I think would be the next sort of, like it wouldn't be a Go, it would be a CO, right? A C annotation where you can start annotating the cell types and the subcell types that are here by like this whole list of functions and processes that already exist. And to use that as like you wouldn't be done, right? But it would give you a context to start performing more experiments, to start doing the unsupervised machine learning stuff on it. So at least you could put it in a context of where you could do it. So that I think that's an effort that could be like extended by this group or other groups.
Absolutely. So hold that thought. We'll come back to Partha behind first.
We began with talking about dependencies of various kinds, and if we look at a set of dependencies and look at inter-individual variation in the sets of dependencies, and if we have a molecular readout of what impact it has on a phenotype, cellular phenotype, even broader phenotype, that probably would also be profitably help us in understanding functions of these dependencies. That would be another way to go, I think.
So at the back, is there a microphone right at the back in the... Matthias, yes, sorry.
Matthias Kassler, Michigan, nephrology, anatomist and physiologist. And I think that's one of the key questions. It's fascinating to see over the last day and a half, cell biologists and computational scientists, that communication is fascinating. And things are clicking, which I would not have imagined to see clicking 8 years ago. But when we are talking about function, who do we talk about to? And what is missing in this audience is the deep physiology expertise. We have heard a little bit yesterday on neurophysiology, that classifying neurons by their physiological excitability is one approach. There's a very rich domain expertise out there, which if we want to reach our next step, to go from cells to organismal levels, we have to talk to these communities. And developing a user persona where our atlas is embedded in the knowledge background of those individuals and where they are coming from. And that's hard work, but really will be critical, and I think we will learn a lot. The caveat is many of these people are at the end of their career, and if we are not careful, I have reactivated my mentor, age 86, to tell us about all these capillary endothelial cells in the renal medulla, and it was enlightening what he could direct us to. So this is really a window in time which hopefully we can leverage and get the excitement in this room to these domain expertise.
Absolutely. So online and then Piero. So down the front for the microphone to Piero.
Thank
online.
you. This is from Seville Oskay Halassi from Hatta. Immune cells have plasticity, so the gene expression program and or epigenetic characteristics are changing depending on their plasticity. How can we achieve this challenge technically to make a reference immune cell atlas?
I can try first and others can comment. That plasticity is part of what they are. You don't try to remove it and then say there's some other, abstract, hypothetical, pure immune cell. That's exactly one of those things that you have these factors that contribute to. In reality, what we see with immune cells, that at any moment in time, you're going to see a whole distribution of them.
Yeah.
And that distribution is actually their characterization. It's not that there's some other thing and once in a while they become that thing. It's just that they span like that. That distribution in its own right shifts and moves and so on. That's one of the reasons that when John said that it's kind of a trivial but not truly trivial question, in which cells is my gene of interest expressed? This is exactly one of those places where it's like, where exactly are my cells, the thing that I'm actually referring to? Once you go to immune cells, it's harder in this way. But it's also quite fascinating, and that's also their nature. We can't make them something that they're not. That's what they are.
So Dana, then Piero, and then.
So plasticity is actually my favorite feature of biology, and this is why I've been advocating quite a lot that we need to go away from just these cell type labels and say whatever CD8 T cell, and really begin breaking the cell down into these gene programs and what the cell is doing. So the immune cell atlas, in my view, is what are all the different states immune cells can be in across different health, diseases, geographic locations, context, tissues. And again, if we're talking about the factors, the forces that bring him there, and the dependencies, I'll use Aviv's terms, okay, so we now have basically catalog of all the different states that the cells can be in, ideally broken down into gene programs, so it's not these million discrete states, but these sort of Lego blocks of programs that a cell can take, and these are quantitative, not discrete as well. And then, why are these cells behaving this way? Are they in a particular context, wound healing, disease, inflammation, viral infection, disease? So to begin to understand what are all the different states that you can have, how do they influence one another, what are the factors of disease, genetics, condition, cell type lineage, spatial location, tissue, that are actually creating these observed immune cells in all these different contexts. That's what the immune cell atlas is, and understanding all the different possible states and why different cells are in these different states is exactly the goal of the atlas.
Piero?
Yes, thank you very much. And relating a little bit to Dana's points of having all those networks to regulate. I was wondering how many different data set or type of data we should still collect. For instance, there is a very important component in RNAs to regulate chromatin activity, to regulate many other aspects of networks. And at least in this project, we're still at, for the long non-coding RNA, for RNA from the repeat element, for many other types of regulation, exchange of RNAs from cell to cells. So we are still very, we are still scratching the surface, and we didn't look in details about that. And there may be many more type of interactions. So shall we start to make a list of types of data that we should still collect to really fully understand how cells are regulated in different contexts, and how to obtain, how to understand what are the fine-tuning molecules, which will include RNAs, but not only RNAs, which will be useful in 5 years from now to really regulate phenotypes, and really using this atlas for therapies where we will need to change gene expression by a few percent in some specific cells, and for this we will need to understand with much more accuracy what happens in all the cells.
Yeah, I think that's a great point. So to go on, then I will move on to the next part of the discussion, but one more comment, please.
Thank you. So I was struck by 2 points, I think, that Aviv made. One is, structure follows function, and you could use supervised versus unsupervised. So structure does follow function. So you need some kind of framework. I mean, there is unlimited amount of things that can be done with the technology, and but if you look at it in a practical sense, what would be the best bang for the buck? At least for the version 1. So there is the extracellular and the intracellular. The intracellular, I think, probably everybody would agree that you need to understand genes and modules inside the cell. The extracellular part, I mean, if you map the whole kidney from the one end to the other, would you, I mean, there might be some differences in maybe gradients of gene expression, etc., but do you really get more value out of it in terms of practical utility versus if you look at it as a functional unit? How does it make urine? I think someone mentioned you look at physiology as the functional unit. So if you look at that, how does the liver make bile? Or, if you look at, we need physiologists to be involved. So if you look at that as what is the anatomical framework, that has to be histology. We need to understand cells in the context of histology, which is the supervised approach, and I think that's a very, very valid approach. So in the brain, you might need, location might be important, in the lymph node, location might be important, but maybe in the liver or the lung, location may not be important. So it has to be kind of, so maybe we should think about organ-specific, what units need to be studied. And then in terms of function, it has to be based on physiology. What does the organ do, and how do all these cells in that neighborhood join together to perform that function as a framework. So structural and functional framework.
Yeah. So I think taking so that comment and everything else we've heard together, in some sense what we need, or what I'm hearing the room say, is that we would like to be able to summarize cells as a function of individual factors, be they gene modules, be they where they are in an organ, etc. We also want to model an organ as a collection of cells. So we have a clear hierarchy here, and then we want to model the whole body or different organs as a function of those organs. And then going to the evolutionary perspective, we want to look at how the cell types and the organs and the bodies have evolved from an evolutionary perspective. This is not a small task or a trivial one. And I would like to ask Dana and Aviv to start by the next part of the discussion by asking, what computational tools do we need to develop in order to address some of this? Is this just a case of applying existing approaches? Do we need to develop new approaches? And are the challenges statistical, or are they more in the computer science the scaling side. At one point, using Chair's prerogative, if I will, at the beginning, as a statistician, one of the things that really gets on my nerves is when we treat a sample as having no error. We often look at Visium sections as though it's just a gift from God and that there's no variability. But if you took another section from the same tissue, just a few microns apart, it would have a slightly different layout of cells. So what we are sampling here is a random sample from a population. So we have to think of this not as a deterministic model, because it sure as anything isn't a deterministic model, but as a statistical model where we're getting a sample from the population. And I think we have to think about it like that. But this is a very strong bias as someone who was trained as a statistician, and we'll now turn to two computer scientists and see if they fundamentally disagree with what I'm going to say.
Yeah, I just love the fact that you organize the world such that statisticians get the model and then the computer scientists will come, and it's just a problem of scale. I found that irresistible to point out.
I mean, machine learning is just a subset of statistics.
It's just a matter of scale, yeah.
Anyway.
I think it's one of, how did, now it's become our motto, everything, everywhere, all at once. Yeah, it's going to be many, many problems like that, not just one. That's definitely one of them. Yes, it's sampling from a distribution. And it's a distribution not just across. There's noise from the measurement itself. There's variation in biology, which is, I will call it, interesting and uninteresting, because I don't think there's any other way of actually parsing those out away from each other. There's the things we happen to care about that we care about is a biological variation, but there's biological variation. Variation is inherent to biological systems. And then there's inter-individual variation, variation across locations and regions. So there's many, many, many of those. And yes, you want to model them well. People mentioned so many problems here, so it's hard to now go, and I didn't take notes sufficiently to now go through each and every one of them. I thought the one that we discussed kind of when Zorina asked her question is one where definitely there's room for new algorithmic work on both the multi-scale stuff, so finding a good latent representation of many different things. It tends to be useful in these settings, and doing it in a way that also in an unsupervised setting can decompose the relevant pieces. I always if you can do it in an unsupervised way, it's always more appealing, because then you can compare to the supervised stuff, say, I caught that, and make new discoveries as well. That's an example. But the question I asked in the beginning about dependencies is a different set of problems. There's something we didn't talk a lot about here, which is predicting. So we kind of take it as a given that if we want something, we have to measure it. But in reality, with these amounts of data, we should be able to train models that liberate us gradually from the need to do as much measurement and replace it with a prediction of a model. If you want to identify, we call them factors here that impact the different aspects of cell states, you can think about first statistical models and then try to go all the way to causal inference to try and really identify hidden causes. There's many, 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 many computational approaches that are probably going to be needed. I think, though, more important than figuring out the computational solution or statistical one, you have to define the problem. And with many, many of those things, the definition, the way that we discuss it, is actually a little too mushy to make it into a method. But defining a good, crisp, clean problem, then you can go and you can solve it. So if there's one important thing is to come up with new problems that we haven't solved yet. In addition to, providing better and better solutions to the problems that we already formulated.
Dana?
So first of all, I really do think that everything, everywhere, all at once should become the new HCA motto. And there's just so many computational problems and, a lot of data, so we're not going to be out of work anytime soon. So maybe that's a good thing. And I completely agree with Aviv that many of the questions aren't well defined enough. Even what is a cell type to do this? I also completely agree with you that it's a population, that it's sampling, that it's statistics. And I think we did a pretty good job at viewing the genes expressed in an individual cell from single cell RNA-seq as whatever. We sampled transcripts, and as technologies improved, we sampled bigger and bigger proportions of these transcripts. I think we're doing a poorer job of it when we're going to compare individuals, when our atlases are going to become hundreds of individuals, and the attitude is, smushing it all into one big UMAP, trying to actually remove the variation. I think right now that's one of the biggest Achilles heels of the field, that, really we lose a lot of the individual variation because many of the harmonization and integration methods that we're using actually actively takes out real biologically meaningful variation. And, that dovetails on what I said last time, that we really need to begin defining sufficient statistics that are well adapted to the problems that we want. Some of these sufficient statistics could be cell states, gene programs, and histomodules or whatever, to begin summarizing the data. I think a critical thing that we need is representation. I think one of the reasons we went forward so well and made so much progress and so much discoveries with RNA is we had a good, decent representation. We had a good idea of picking out meaningful features, highly variable genes, maybe enriched for some of our favored biology if we knew of them. We had a good representation and metric of similarity, the cell-cell similarity graph, the phenotypic manifold, and because we had a good representation, we made so much mileage. I think we're not in a good position to have good representations of attack data, and Christina made some attempt to show some initial attempts at that, but I think we're trying to copy the single cell data and do the same for attack, and we're missing a good representation there, certainly one that will get regulation, causality, and things like that. Spatial methods, again, we're defining these neighborhoods as discrete cell types. We don't have a good representation to even begin addressing all the questions that were raised here. So I think sufficient statistics, representations, and even defining the questions is the first step before you go build fancy methods. But some of my favorite methods to build is, again, beginning interpreting the spatial data of doing multi-scale, and multi-scale is now at a spatial scale, models of how cells begin to come together for tissue function. One of my favorite problems, which I think we're pretty weak at computationally, and while and gene programs and factors, and I think we've made some mileage on factor analysis, but we're still at the very, very, very beginning, as I defined at the beginning. Let's break down what we see in a cell as gene programs and the factors that make these gene programs what they are. So those are my two favorite big problems. They are interrelated. Everything is dependencies. And for this, we need statistics to treat this as population, to understand rather than move the variation under the hood, sufficient statistics and representations.
Great. And I would just say that for those playing Dana Bingo, smushing everything together into a UMAP just came up. So for those who've playing bingo, you've won. Emma first.
So there was a lot of talk about physiology and function, and I think that this is maybe an underappreciated area where methods or computational approaches could be taken. But Aviv was saying we don't need to measure everything, but I wonder, do we actually maybe need to pair, or at least have some subset of data where we actually pair some physiological measurements from clinical type of approaches? Because even I was struck by the disease talks that we saw yesterday. We always label things as, you have a chronic liver disease or you have Alzheimer's, but actually the interesting findings were correlating things like amyloid plaque deposition or, size of the necrotic. So very specific physiological measurements, and there was also physiological measurements in health or traits that are closer to the molecular phenotype than the complex disease, but we don't really collect that a lot. So do we need this?
I would say absolutely, and what's interesting about it is that it's bespoke. So I think for this community, people are used to actually not having to rely on bespoke assays, right? It doesn't matter what it is. It can be it can be genome sequencing, it can be single cells, it can be your favorite spatial method. It's not bespoke in the sense that you take the same approach, you apply it here in the body, there in the body, it's always the same approach. Whereas for physiology, and I think that was also the point that Matthias was trying to make, I thought he was sitting over there, was that it's actually quite bespoke. And so in Alzheimer's, yes, you would go with A beta and a tau. And by the way, you go after them because you already understand them. They're functional proxies. You understand them and they're causal. And you know kind of the causal chain. That's what Phil could do in his work. You don't always have that privilege that you have something like that, but when you do, you need that physiological measurement. And here you start getting into all sorts of trouble. For example, we rely on invasive assays. We actually need samples from inside the human body, so they don't kind of naturally come also with physiological measurements at the same time. What works so nicely for Phil's study is that this is a prospective cohort that is collected when people enter that cohort, if I remember correctly, at 60 or 65, and they commit to participate until they pass away, which can be 20, 25 years later. And so yes, at post-mortem, you get their A beta, their tau, their this and that. You get cognitive measurements every year. But you actually have to do it prospectively. So we're kind of in a less than ideal situation like that. So that's where it becomes interesting, for example, to be able to make predictions. But because it could be that you could get your physiological measurement in something pretty mundane, like histology, in one setting, and you can get histology and molecular information in another setting, but you wouldn't be able to get all of them chained together. So you have to find a computational way of actually bridging between them. But yeah.
One comment down here, and then we'll move on to a second part.
I actually always agree with Aviv, but there.
Dangerous.
I just want to say that's a great idea. I wish my kids would also subscribe to that.
Okay, so I think that for me, practically, this comes down to like metacell and metagene. And so if we're talking, if you're going to make a metacell, then what do you want, what metadata do you want about it? You want to know everything about the sample that it came from, and including the the position, the disease attributes of the donor, all this, the whole provenance of the what led this biological thing to be formed and as you sample it. And so, that even includes the technology that was used to measure it. If you've got multimodal technologies on that cell and that location, they have to share attributes that let you get the grouping and make easy connection of those two meta cells and the features of that meta cell. And then you get, and how many are there of that, and what were is it all in this location, or now you're bridging multiple locations that are getting you this meta cell that that has all these characters. So everything on top of that bin, it's almost like a bin for the meta cell, and we're very undisciplined about the way that we form those. We use the different values that is if it's 100 cells and they all have these varying levels of counts of feature 1, 2, 3 in them. Or should we, for all the cells in that group of 100, be used equally to form that estimate of the value of the feature that's measured in it, or should there be another approach so all of these things are really change the way that data is used, the power of the data that comes out of that meta cell. But if you don't have that metadata about everything about spatial and do you think that this cell belongs to a group? Was there a treatment that was up behind it? All that stuff can go into groups of metadata that are in that bin, and we just need to be very disciplined about it. Okay, now think about like the measured features. So a gene list, and so now it's like a metagene, and I know this causes Donna pain, but there's gold in them that that subsumption, and say, look, these are all the freaking ribosomal protein genes, and they always behave the same, and unless you're in a very pathological circumstance, just make a freaking metagene out of it. But then for the cell cycle, are we disciplined about is this an S phase, is this an early S phase, is this a G2M phase cell cycle? That's useful. We don't have that metadata about a gene module now in a very understandable, standardized way. It's such an easy thing. It says, is this an mTOR pathway module? Is this a module that you just observe in your data that's in this, under this context? Great, label it, have that as a feature that's now, part of the supplementary data or something that's in, in your data set that was your criteria. There's another level too on hierarchies, on meta genes or meta features. And something that sometimes, like when we think about, okay, these are all the fibroblast stromal, these are all the myocytic stromal cells. You're actually using a group of genes to do that are working across multiple cell classes. So say that, this is a level 1 hierarchy gene module that is similarly regulated across these cells, these states. Here's now the next substate below it. That's a level 2 hierarchy module, and then same for this end state that you think is a singular. Those modules are very useful to combine to make a composite system view of the the networks that are operative in that cell that are shared or dissimilar between classes and subclasses.
Yeah, I would say it's not so trivial to know some of these things you've got to infer it, but that's a separate conversation. But I would like to move on to a second thing, which we haven't touched on yet. We've focused a lot on what to do with an atlas. Now I'd like to move on to a second question, which is what to do when you have a new data set from a disease context, and how are you going to interrogate that in the light of the Human Cell Atlas? And many of us have seen this. You have a new data set, you think, great, I'm going to integrate it together with a reference atlas, all good. Oh, but they don't overlap quite as much as I'd like. And is that biology? Is it technical? And the answer is often, we don't know. But the way to think about this perhaps is, we're very obsessed, and on this I agree with Dana. We agree on lots of things. We just like to disagree in public, sometimes it's fun. But for many papers, figure 1 is a UMAP of everything. It's the smushed together UMAP where we put together all our samples so we can display it in one figure, and that's kind of how we start. And that's whether we have a disease sample we're comparing, trying to combine with a reference or whatever. Is that actually right though? Do we actually have to integrate them onto the same manifold? Or can we think about other ways of interrogating new samples from a disease context, from new biological samples that don't require it all to be integrated? And so I just, as before, have a series of questions that I think one might like to ask of a new sample in the context of a reference that don't necessarily require them to be integrated. So, very simple. Are all the same cell types present in my new sample as they are in my reference? Sounds simple, not quite so simple, of course. Do I have similar sets of gene modules expressed? Are the same gradients present? So similar to the previous question. Is the overall transcriptional profile, however one wants to define it, overall transcriptional activity, number of introns that are sort of the number of pre-RNAs, etc., are they similar? Now, turning to Dana and Aviv first, do we actually need to integrate the disease or our new samples together with a reference to answer some of these fundamental questions? So maybe starting with that, and similarly, as before, what are the other types of questions that one might want to ask of a new sample in the context of an atlas that might not require integration? So starting with Dana first and then on to Aviv.
So of course my answer is no. I think that sometimes integration loses information, especially because we're so bad at distinguishing batch from biology. And again, I'm beginning to sound like a broken record, but that's why we need to begin to define sufficient statistics, whether it's sort of metacells / cell states that are very, very sort of tight and defined. These could be defined by a set of quantitative gene programs that define what they're doing, but really breaking it down to sort of entities that also have biological meaning. These gene programs and cell states have a role. And I do think that these entities, once you begin aggregating, if you begin aggregating genes together into a gene program that's more robust than an expression of individual genes that you're trying to integrate in individual cells. If you aggregate multiple cells in the same state as a metacell, a concept that Amos Tanai first brought, again, you have huge statistical power because now you've sampled 100 cells in the same cell state. And now you have a better grip on comparing batch and biology. First, just the aggregation itself protects you a little bit from batch. The aggregation, especially if you're beginning to have interpretable features of why these gene programs are active, what they're doing, also allows you to sort of understand data, understand what's different between disease and normal. So that's why I think we really need to push towards these aggregated sufficient statistics, again, depending on our question. I think not only are the same cell types / cell states expressed, and I think sometimes you might see slight shifts in these rather than completely different cell types. So usually the answer will be yes, same cell types are expressed, except we see slight shifts in their gene programs. That's what Milo always shows, your wonderful algorithm. The proportions of the cell types, wildly different. You can have even the exact same cell states in slightly different proportions. That's what creates a lot of disease, certainly in the lung. And of course, one sees that one of the biggest things that's disrupted in disease, and Paolo showed beautiful examples of that today, is the spatial organization of these cells. And now that spatial methods are coming online, now that there are actual commercial usable technologies where we can collect spatial data, seeing how the actual organization of cells, which actually drives what they're doing and how they're interacting and influencing each other, is one of the biggest differences in disease. And once we can also, if we have these sufficient statistics and these contexts and factors that actually have meaning and causality and explanation behind them, not only can we mitigate batch versus biology, but we could get insight into what's going wrong.
So just to push on that, and then I'll come to Aviv. So what you're proposing, just to be clear for people, is that one could compute a sufficient statistic independently on
multiple
the reference.
Types of sufficient statistics.
Yeah, of course, not just one. There's not going to be one sufficient statistic. But you could compute a vector of sufficient statistics independently for two data sets, and you could just compare those, and there would be no need to formally integrate, and that's how you would effectively discriminate between the two in fine signal. That's what you're proposing, Dana?
Yes, I would actually do it at the individual level. So even within a data set, like I would actually create these sufficient statistics for every individual in our healthy controls. I would create sufficient statistics for every individual in our diseased individuals, and basically the sufficient statistics would be on a single individual so that we can capture the variation, because each individual is different and even each disease state is different. And that also helps us sort of overcome batch. So I would basically take each individual that we collect, each individual sample, if we take multiple organs from this individual, and then collect a whole battery of sufficient statistics, which I threw a couple examples, but I think we only began to define what sufficient statistics we can collect. And I think that as we see how these work, and we collect bigger cohorts of individuals, and we crystallize our questions. I agree with Aviv that we're terrible at formulating very accurate and precise questions. The sufficient statistics that we will need to actually address them will grow, but I think we need to switch from these smushed, fully integrated UMaps of individual cells across multiple data sets and individuals into sufficient statistics. That's the field has to go as we scale. It also helps with scale, because these sufficient statistics are much smaller than, whatever, 100 million cells.
So just one thing, and then I promise I will go to Aviv. Is this an argument against ever integrating data? At some point, once you know the sufficient statistics to compute, do you actually ever need to integrate anything? Because you've got a per sample, you could just compute the statistics per sample. So is there any need? Are we arguing ourselves out of a human cell atlas in some sense, the classical way we're thinking about it, if we go down the road that you're arguing?
I think the human cell atlas is the sufficient statistics. I think this is a better way to go than integration. Of course, if you don't integrate and put that pretty UMAP, the reviewers won't publish your paper. But but I actually am, guilty as charged. I started with integration, but as I saw the problems with integration, as I saw the real biology lost when integration in the C cells paper that I published, we really see how all the standard integration approaches actually remove disease progression in COVID. As I saw the flaws of integration, I'm beginning to sort of move away from integration altogether in the way I think about the data.
Aviv, no need for a human cell atlas?
I wasn't.
A different cell atlas.
No, I think, first of all, I would say the word integration is, again, not well defined, just to be fair. So to me, that's not the question. It can be implemented in many different ways. So I don't have any particular attachment. I think most people, when they mean integration, they mean something that cleans up the batch effect, that makes things look similar to each other. To be fair, that's not necessarily the best approach. In some cases, it can be useful. As a minor comment on the sufficient statistics, I was not going to talk about them at all, is that they actually do need to be eventually sufficient. And actually getting to what is sufficient but minimal, and this and that, it actually gets a little difficult. They can be sufficient for something, not insufficient for another. So that's as a side note. I don't actually want to talk about them. I'll say two things. The first is that integration is not the only way of querying something. There's many other ways to query something. For example, if you have a good metric, and the metric is robust enough, you might be able to go and query something without first taking the new entity, sticking it inside the other, and comparing. We query genomes like that all the time. Of course, there are some things that are easier for us. There is a problem, but it seems very natural for us to do it that way. And there's many other things where you can do querying. So that's also not the most, necessarily the only approach. I'm kind of a pragmatic person. I usually think there's multiple ways to solve, and often when there's multiple ways to solve, they provide different solutions that are useful for different things. So I don't love making an upfront statement saying, let's not do this and only do that, because it turns out for this one approach could be quite useful, and for that, the other approach could be quite useful. Second comment I wanted to make is that I believe you also asked us what other questions might we ask. So I wanted to say something about that. We're getting close to the end of the session. There's a parallel session actually running on foundational models. So I feel maybe pushing a little bit in that direction, but in particular to highlight the importance of the literature. So the way you framed for us is that you said you have a reference and you have your new data set. And there were all sorts of assumptions there. The first one was that I can only use the reference and the new data set. Somebody mentioned the Go terms earlier. I would say there's a rich biological literature. Not just Go terms. There's like papers upon papers upon papers. And today what we do is that, in fact, we put everything together, and then we get something, and then we get some genes, and then we go and we search PubMed. That's actually not how the future should look like. The literature, all that rich information about cells and genes and physiology and this and that and the other, should be part of the process. So for example, imagine you got this new sample. It was maybe dissociated, maybe spatial, maybe multiscale. It doesn't matter. You did a bunch of measurements on it. And you now want to query a big atlas. And it can have reference samples. It can have disease samples in it. What if that thing is a tumor of unknown origin, for example? You don't even know what to compare it to. So wouldn't it have been interesting if I could kind of take my new sample and I would say, what out there looks like this thing? Rather than me assuming up front that I'm comparing it to the right reference. Just tell me what it is. I mean, when I take a sequence and I blast it, I don't actually usually limit my search space, because it seems to me completely reasonable that the best answer will come back to me through blast. So that's one layer of it. And the second is, why can't the literature be a partner in this process, both in guiding what the best match would be, but also in telling me what it might be about? For example, it might tell me it's cancer, and it might tell me, well, given these cells and these genes and all of those things, these processes might be going on. It might be an infiltrated tumor, because there's a lot of T cells, and infiltrated tumors, they have particular outcomes, to Emma's earlier point. It could actually bring all this historical knowledge also for the ride, and to Dana's point, maybe some hallucination that would make me make better predictions. So that's to me kind of a more ultimate goal. Might take a little bit of time, but all these people were in this other session on foundational models. Maybe they solved it by now.
Absolutely, I'm sure they have. So we have to wrap up, I'm afraid. So I just wanted to take a minute or two just to sum summarize this discussion, which I think has been really enjoyable. And thank you again to Aviv and Dana for driving so much of this and being such willing participants. I think a few themes have come out of this. First, that there are multiple ways to interrogate an atlas, and we have to work on all of those. There's merit, definitely, in the integration, especially for the identification of rare cell types. The ability to really nail those down requires the combination of data across individuals. You're just not going to have the power otherwise. But we also need to think about other approaches, sufficient statistics, whether it be a way of characterizing at a per cell level the set of modules that are expressed, the gradients that are active within a particular cell type that are present in a particular region of the data. And we have to be able to compute those, because those are going to be the things perhaps that we want to query directly with new data sets rather than formally integrating everything together. And a third way, which was discussed in the parallel session that Fabian was leading, around foundation models, how we put this data together so that we could then begin to reasonably hallucinate to understand regions of the space that we're not able to sample well. And that's a way of bringing together some of this physiological data that we talked about, that's so important, some of the literature. That's where I personally think that it's going to be the easiest to incorporate some of these data in the foundational models, just because they are much more flexible in structure than the sort of classical statistical approaches that we think of from a regression perspective. When it comes to classical sufficient statistics, I suspect that just the challenges of data formats, et cetera, are going to be just much more amenable to looking for correlations in large-scale foundational models. And if we do this right, this is going to give us multiple views of a human cell atlas. We're going to be able to summarize data in hierarchical and organized way, from cells to organs to organism, across organism, and we're going to need all of these views. And I think as a computational scientist, as a statistician, it's tremendously exciting, because we're really scratching the surface of developing these approaches, and we have to work together with our colleagues on the bench, physiologists that we've heard about, to really frame those questions and then come up with the approaches, come up with the computational tools, both accelerating the way that we access data, but also the new approaches, the new ways of summarizing that data, linking the sufficient statistics, the foundation models, and the integration. I think by so doing, we're going to have a useful human cell atlas, and ultimately, that's really what all of us want. So thank you all for participating in this session. Enjoy lunch.